Hello everyone, welcome to our video lecture. So students, in our last lecture we have studied about configurations like DL configuration. Okay, DL configuration, if I say, it was studied mainly for carbohydrates and amino acids. Okay, so let's continue our topic. Now, relative versus absolute configuration. So first of all, question is, what is a relative configuration and what is absolute configuration? Okay, so we have studied DL configuration and we have studied RS configuration. Okay, so DL configuration, if I say, it was studied mainly for carbohydrates and amino acids, right? And also it was relative in the sense, means suppose if you say that for a particular amino acid it was specified with respect to diglyceraldehyde. First of all, remember diglyceraldehyde, top portion with aldehyde CHO, down portion with CH2OH, right side with OH and left side with H. So with respect to that we said right side OH, then it is D form, and with respect to that, relative to that, left side will be L form. So it was our relative configuration. Okay, clear? So it was relative configuration with respect to, particularly derived with respect to diglyceraldehyde, and also for carbohydrates and amino acids it is studied. Next, relative study and with respect to diglyceraldehyde, all other forms are studied. Okay, so it was mainly first of all discussed with respect to glyceraldehyde compound, D form, then its L form mirror image, then with respect to that further. Okay, now if I say DL configuration, then this capital DL configuration has no relation with small d small l. Okay, you can say, I am just writing here, no relation with small d small l. Okay, small d small l wal experimental dextro and levo. Now let's come to RS configuration. What RS configuration says? See, I'll just compare it. No relation with DL. Yes, for RS configuration also, it was just the two names, rectus for right side and so sinister for left side. Okay, rectus for right side clockwise. We just mentioned with respect to priority CIP rule. Okay, so it has also no relation with DL, right? R configuration may be small d or small l, it may be dextro or levo. Okay, relative configuration, yes. Now why we say RS configuration as an absolute configuration? So students, you need to understand, for RS naming, you remember we studied entire rule, just like IPSC naming we studied. Similarly for RS naming we studied the rules, CIP rules for RS nomenclature, for RS configuration defining the RS. Okay, so it is absolute for a particular compound, it has no relation with any another compound. Absolute configuration. Now for carbohydrates and amino acids, it is not a specific to any particular group or compound can be studied for every chiral center. See, for every chiral center we can define whether it is R configuration or S configuration with respect to the predefined rules. Okay. Now, relative study. It is absolute study. With respect to diglyceraldehyde, all other forms are studied. Now, with respect to diglyceraldehyde, it was studied mainly. Okay. Here, for all compound having chiral center RS is defined. Okay students, so for all compounds which are having chiral center, RS will be defined. Clear? So let's see a few examples to differentiate these two and also to study in carbohydrates about RS configuration. Now you have to find RS configuration in given compounds. Okay. Okay. So dear students, you can see these two compounds. Here you have to find out RS configuration. Okay, I am drawing here one more case. Okay, so I think you must have completed these two cases. Let's check one by one. So first of all, in this question, if I say it is having aldehyde part, alcohol part and this HOH. So it is having one chiral center, and as we know, if a compound is having chiral center, we can define RS configuration. So first of all, let's check the priority order. Here carbon, oxygen, carbon and H. So oxygen will be 1. Here this part, as we can say, it is C double bond O and H, means carbon is connected with 2 oxygens hypothetically. Here carbon is connected with 1 oxygen. So you can say this will be second priority, this is our third priority, and this is fourth priority. Now, as it is anti-clockwise, so using shortcut SARC, we can say S configuration, but lowest priority on horizontal line, so it is R configuration. Clear to all of you? Okay. Next, let's check this one. In this one, if I see, then this is having oxygen, carbon, carbon and hydrogen. So oxygen will be first. Okay, now check this one. This carbon is having oxygen, hydrogen and carbon, and this carbon is having 3 oxygens, as it is C double bond O and O. So 2 here and 1 here. Okay, so first of all this OH and then this one. So it is anti-clockwise S, but hydrogen in horizontal, so it is R configuration. Okay. Now if I say this as numbering 1, 2, 3 and 4. Okay. Now if you check this one, then 2R. Let's see third. This OH will be first priority, obviously, oxygen. Now carbon, hydrogen and carbon. Carbon is common both sides. This one carbon and this carbon. Now this is connected with O, C and H. Now from this, carbon is connected with 2H and 1O. So this upside will be priority for this OH. So it is also anti-clockwise, but this hydrogen in horizontal, so it is clockwise, so it is again R. Clear to you all? So I have checked this configuration. 1, 2, anti, reverse, R. This one 1, 2, anti, reverse, R. Okay. Let's check that one. From this, if I check, This oxygen will be 1, this will be 2, 3 and 4. So it is what? Clockwise, but hydrogen here in horizontal, so it will be anti-clockwise. So configuration is S. So dear students, you can see in these three questions, this compound is having R configuration, this is having R configuration, and this one, if you see, this one S configuration. Now, let me define and compare RS versus DL. Check this one. This is what? This is actually OH right side, so it is having D configuration, okay? It is your D-glyceraldehyde. Check the formula. It is our D-glyceraldehyde, okay? Upside functional group, this formula. And this formula, if you remember, actually 1 and 3 are mirror image, if you see. So it is our L-glyceraldehyde, okay? Clear? So this is D-glyceraldehyde, this is L-glyceraldehyde. Done? Now, there is no relation between DL configuration with RS configuration. One more part, okay? Now let's see this one. Whether this is carbohydrate part or something like that. Yes. So when we define perfectly carbohydrate, we say hydrates of carbon or aldehydes or ketone having functional group aldehyde and ketone having polyhydroxy. Here you can see that this compound can be generated when? This compound can be generated now look at this compound. If I say HOH, HOH, CH2H, CH2H common, only this part. If we take any such reagent which can oxidize this aldehyde part without disturbing any other part, then we will get this compound, okay? So in this compound also I have defined RS configuration, but if we say simply the D-glyceraldehyde derivatives part, then I can also give its configuration here, right? With respect to this main functional group at the top and carbon chain in vertical line and this second last OH right side, so it is D configuration. You remember, it will be D compound, okay? Just like this one, D configuration, okay? So students, I think all of you must have understood in detail about this case, right? How to check DL configuration, RS configuration. There is no relation between capital DL with small d small l, no relation of RS with small d small l. Clear? Okay. Okay, so students, let's see and understand the rules defined for DL configuration and how to check in such compounds. Generally, the question is asked mainly for D-glyceraldehyde. So let's see the application of even and odd interchange here. Okay. Now here, what I need is, I need this main function group at the top. Okay. So to check the DL configuration, let's do even interchange. So I will get the identical compound, right? So if I first of all interchange this aldehyde and hydrogen part, what I am going to get? This will be as it is, CH2OH. Now this aldehyde CHO part comes upside, okay? Now hydrogen comes here, first interchange. Now hydrogen and OH will be interchanged. So OH comes this side and hydrogen comes this side. Clear? Okay. Now let's see the configuration of this one, whether it is D form or L form. So what I find is top function group at the top, main function group, and as I say second last OH. See, sometimes it is also said that the last chiral carbon with respect to the high priority functional group. It may also be considered same rule, okay? Whether you say second last OH or you can say the last chiral center with respect to main function group which is at the top. It is not necessary it will be on the top. It may be on the top side, okay? Okay. So you can say it is left side, so it is having L configuration. Clear to you all? Now let me check whether I have done correctly or not using RS configuration here. Just to check. See, this OH will be 1, this 2 and this 3. 1, 2, 3. This is anti-clockwise. So it is having S configuration. As I know, by interchanging the configuration must remain same. Okay, let's check this one. OH1, this is 2. This is your clockwise, so R, but hydrogen here, so it is having S configuration. So my interchange is also correct. Okay, so in this way you can use RS for finding DL. For the examination purpose, to make yourself easier side, you can use this logic. Let's check this one. See, here I need this main function group at the top side and this group at downside. Okay, so one possibility is I can make this interchange and this interchange. Okay, so let's see this one. One interchange in these and one interchange in this. Okay, now let's check RS configuration to assure whether we have done correctly or not. Now this oxygen 1, this is 2. So you can say it is anti-clockwise having S configuration. Clear? This is S configuration. Hydrogen at the vertical line. Now check this one. 1, 2. Now it is your clockwise R, but hydrogen here. So it is S configuration. So this even interchange is possible. Okay, as I told you earlier also. So this OH is left side, so it is having L configuration. Okay friends, so all of you must have completed and understood 100% about this part. Okay, so let's study further topics. Apimers. Now what are apimers? See, this is a term which is used in various cases. Like if I see biology students, then they may come across this term in biology biomolecules chapter, okay, in standard 11 syllabus, if I say about NCERT. Now if I say about standard 12 chemistry, there is a chapter biomolecules in bio as well as chemistry, both same, biomolecules. In biomolecules also you will come across this term apimers. Okay, now what is apimers and what is the use here? So if I say apimers, it is all about the diastereomers having, see, two optical isomers of each other which are diastereomers such that they differ between each other with respect to only one chiral carbon. Means the compounds which are having different configuration across any one chiral carbon only, rest all configurations are same and they must be diastereomers of each other. Okay, so if I say apimers, diastereomers which have different configuration across any one chiral carbon and all other parts remain same. Okay, so they must be diastereomers. Obviously, if in two compounds, if I say across only one chiral carbon, means it definitely says there must be two or more chiral centers, and out of two or more chiral carbons, I am saying only configuration across any one chiral carbon is different. So when you say about mirror image relation, they won't bear any mirror image relationship. Also, they won't superimpose each other. They are not superposable. So they must be diastereomers of each other. So let's see examples. Okay, so students, look at this compound carefully. It is having 1, 2, 3, 4, 5, 6, C6, H, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, H12, O6. Okay, now I am telling you a specified part. This is actually glucose. Okay, relating to 10th standard, all must know that sucrose on hydrolysis gives glucose plus fructose, and glucose and fructose both are having same molecular formula C6H12O6, but they differ in what? They differ in functional part, functional group part. This is aldehyde, that will be ketone, and one more part, this is having different configuration across this carbons if I say. Okay, so let's see about glucose. This is actually your D-glucose in a specific sense. See, functional group at the top, OH at the right side. Okay, see, last chiral center having OH at right side. Clear? Now I have to discuss about epimers here. So this is our glucose. Also for memory, you can recall this all times. Remember this thing. Glucose will be having aldehyde at the top, CH2OH at the bottom, and 4 HOH groups, means 4 chiral centers. Okay, all having HOH groups. Now how to remember? Third OH is on left side. Third OH on the left side in glucose. CHO, HOH 4 times, then CH2OH. Third OH, C3 having OH in the left side. This is glucose. Now, if I draw one more structure, that will be actually your mirror image of this. Okay, now let's see this one. What is the relationship between these two? Are they epimers of each other? No. Why? Because if I see this one, this configuration is suppose R, then it will be definitely yes, because this part and this part is same, only HOH is interchanged. Actually these two are what? These two are enantiomers of each other, as they are mirror image. Also, with respect to main function group, this is your L-glucose part. Okay, now if I say this is 1, this is 2, then 1 and 2 are enantiomers. Now I am just drawing one more case. Okay, this is third one. This is also your C6H12O6. Okay, but this is not a glucose. Okay, that may be some compound having this molecular formula only. Now let's check the difference between 1, 2 and 3, or to check the relation between 1, 2 and 3. Okay, so I can say 1 and 2 bear mirror image relationship, so they are enantiomers. 1 and 2 enantiomer. If I say two and 3, now let's check 2 and 3. Same functional group, bottom same, OHH. At C2 it is same, at C3 it is same, at C4, yes, at C4 it is different, C5 same. Means across this part, if I number 1, 2, 3, 4, 5, 6, this has 1 2 3 4 5 6, and this one also 1 2 3 4 5 6. Then in 2 and 3 I find that across C4 it is having different configuration and rest all are same. I need not to check RS configuration in all these 4 chiral centers. Okay, I can just say across 4 it is different. So 2 and 3 epimers. Also in more than one character type, 2 and 3 are diastereomers. See, if a compound is epimer, it must be diastereomer. But if a compound is diastereomer, it is not necessary that it will or will not be epimer. Now let's check 1 and 3. See, across C2 is different, across C3 it's different, across C4 it is same, across C5 it is also different. Okay, so they 1 and 3 they won't bear epimer relationship, but 1 and 3 if I say configuration across C2 is different, across C3 is different. See, you can check this one and this one. C2 different, C3 different, C4 is same, C5 is different. Okay, across 2, 3 and 5 it is different. So definitely they won't be mirror image with respect to each other. Clear? Okay. So they are diastereomers. They cannot be mirror image and they won't superimpose among each other. Clear to you all? Okay, so students, it was all about epimers. For any given example, you can check this thing. Epimers means across only one chiral carbon, configuration will be different. Rest all things will be same. Okay. Anomers. Now when another term comes, what is anomer? See, anomer means simply it is mainly a part of biomolecules, but I am discussing here because they also bear epimer type relationship only. Okay, what happens? Suppose one open chain compound having some functional group is present. Okay, now that compound can form its a ring type cyclic structure. Okay, I'll explain you with an example of glucose. In glucose, what happens? There occurs some intramolecular reaction and hence that glucose bears cyclic structure as well. Now, during cyclic structure formation, what happens? The carbon which was earlier main functional group, it becomes a chiral center. Now, when main functional group becomes chiral center, when it converts from open chain towards ring formation, then it will bear chiral center. So it can be R form or S form. Okay, so such two compounds formed will be called as anomers. Let me explain you with example first. Okay, as you remember, this is glucose, 4 chiral carbons, third in left side. Now what happens? This oxygen will attack this carbon. Now someone may think how this much distance, in this much distance, how this attack will be possible intramolecular. So in 3 dimension, when we use Fisher, actually it is 2D, right? Sohars and Newman were 3D, but Fisher is actually 2D style. But if you check it in reality, what is this? This is actually in this style. See this one as main function group. Suppose if I say, okay, 2, 3, 4, 5, 1, 2, 3, 4, 5. Now this one. Now you can think yourself. Now this distance is not too much far. Okay. Now, so when this will attack, what will happen? This oxygen will form bond with this carbon, and this oxygen, this bond will break into O minus. Now when it becomes O minus, this hydrogen from this part will go here. Okay, now you check this carbon. This carbon will be having hydrogen, one part, this group, second part, this O with H, third part, and this oxygen bond, fourth part. Means this carbon will be generated as a chiral center. Okay, now when it will be generated as a chiral center, we have two possibilities. Let me draw first alpha inside. Okay, now when I say this will be inside alpha, when I say alpha, see this is your glucose or D-glucose. So when I say alpha D-glucose, then I means cyclic structure of that glucose where the chiral carbon will make OH inside. Okay, now this part inside H. See this is 1. Let me make the numbering. Now what will happen here? This is your first carbon, right? 1. This became chiral now. Now 2 will be as it is because we are not doing any changes here. Third will be as it is. Fourth will be as it is. Fifth. See, in fifth what happens? This side is H and this will be represented like this. Okay. Let me make numbering 1, 2, 3, 4, 5, 6. Now you can see this first carbon was having functional group. When it became cyclic structure, then that functional group carbon became the chiral center having H, OH, this oxygen, this side different part and this side is different part. Okay. Now when we made this OH inside alpha, in simple Hindi terms to recall, this you can say as alpha. It is under. Okay, inside part. Clear? Now you can check remaining part is same, means configuration across second, third and fourth is same as it is. Okay, and see this peak point and this peak point or this peak point, it is not having carbon. Remember, generally we use such peak points for carbon atoms, but here it is just a method of representation. Okay, now this is your alpha D-glucose. Let me draw another case. Now what will be the another case? Another case will be simply when that OH will be outside. Means this OH will be this side. Now this is carbon number 1, 2, 3, 4, 5 and last will be your CH2OH. Across C5, this one, right? Now fifth, fourth will be, see first one, second one is H, OH, third is OH, H, fourth is this OH, H and fifth is H this side. Now you can see this one as this part as beta. Okay, beta means outside. Clear? Means that OH will be outside, so it will be considered as beta part. Clear? Remaining part second, third, fourth are all same. Across fifth we have made. Clear to you all? This will be called as beta D-glucose. Beta is beta means bahar side, beta, and this was your alpha means under side. Okay, so I think you are clear with this. Now what is the application here? When D-glucose exists in this cyclic form, in these two forms, alpha D-glucose and beta D-glucose, then when in open chain compound the carbon was functional group, in closed chain or cyclic structure that carbon atom becomes chiral center. Such two compounds bear relationship called as anomers. Means the relation between, suppose I say 1, 2 and 3, then 1 and 3 are anomers. Okay, clear to you all? 1 and 3 will be anomers. Done? Okay, let me define the definition. Anomers. So if I define anomers, then I can say anomers are cyclic epimers which are also diastereomers having different configuration at that chiral carbon which is main functional group in open chain structure. Okay, you can check the structure. In open chain structure, glucose bears the main functional group. Okay, now when it becomes cyclic, then anomers are cyclic epimers. Okay, diastereomers having different configuration at that chiral carbon, means CHO was your main functional group and that becomes chiral carbon, and with respect to that chiral carbon we have made two forms, alpha form and beta form. So these two bear relationship called anomers. So all anomers will be also epimers only. Why? Because they are having different configuration across one chiral carbon only. If I say specifically, they are studied in carbohydrates actually. Carbohydrates across hemiacetals or hemiketals. Okay, this is no need as such, I am just defining here. We will discuss later in corresponding chapter. Okay, for here you just need to know anomers are diastereomers. Second thing, across one chiral center they are different. Okay, now another part of anomers will be that they are diastereomers. Clear to you all? Okay, so I think all have understood. One more thing I can say, generally it is said that anomers are C1 epimers. C1 epimers in aldehydes or C2 epimers in ketones. Generally, example I can say C1 epimer for glucose. As we know, glucose is having first aldehyde part at the top, first carbon. So across C1 it is anomer or epimer. Now if I say C2 epimer, it is actually about fructose. Okay, so I think everyone must have understood in detail about anomers. So let's continue. Let's study the compounds having two non-identical chiral carbon atoms. Now what is specified about that particular thing? Let's understand. I am taking an example of a compound, suppose CH3, Br, Cl, again this is CH3, H and H. Now students, you can see here that this particular compound, okay, this particular compound is having two chiral centers. This, if I'll make numbering, I can say 2, 3 position is having substituent. Now from this side chloro, 2 chloro and 3 bromo. From this side bromo and chloro. So B comes first. So 1, 2, 3, 4. Okay. 2 bromo, 3 chloro and butane. Clear? Now here if I check the configuration, then this bromine will be first, then downside will be priority. Clockwise, reverse, S. Now this one, 1, 2, anti-clockwise, reverse, clockwise, R. Now students, what happens? If you remember meso part, what was happening? If this was your chiral center having S, this was your R, and it was bearing mirror image relationship, it was meso, plane of symmetry was present. But here, no as such plane of symmetry will be present. Why? Because this one is bromine and this one is chlorine. Although it is having S configuration, but the amount of rotation caused from C2 will be different as compared to C3, because this is chlorine and bromine, both different atoms. Okay, clear? So this compound must be having some sort of optical rotation. Okay, let me say theta. Clear? Now this is optically active compound. So definitely I can draw its enantiomer. Okay, now this is its mirror image relationship. Clear? Now this will be definitely 1, 2, anti-clockwise, reverse clockwise, R, and 3 will be S. Clear? S becomes R and R becomes S. Now this is structure number 1. This is second. Now I have two more possibilities. What can be done? I can draw two other optical isomers of this compound. How? Here I have kept both the hydrogens on same side. So by mirror image it is also having same sides. The two other possibilities include now if I draw this one, then here hydrogen and hydrogen on opposite sides. So this is also an optical isomer. As we say, Fisher, when we observe Fisher, it is actually 2D, right? So here the placement or spatial arrangement is different. Okay. Now, as this is optically active, let's check the configuration. 1, 2, 3, 4. This will be 1 across 2, clockwise, reverse, S, C, 2S as it is. This will be obviously S configuration. Now when I draw its mirror image, I will find its mirror image becomes CH3, Br comes this side, H comes this side, H here, Cl here, CH3. Let's check its configuration. 1, 2, 3, 4. As it was S, it will be R. It is S, this will be R. Okay. Now let's number this as third and fourth. Okay, so dear students, we can say that this particular compound 2 bromo 3 chloro butane is having 4 isomers, 4 stereoisomers, and all these 4 are optically active, right? 2 non-identical chiral carbon atoms. Clear? So 1, 2, 3, 4 all are optically active. Now if I say relation 1 and 2 enantiomer, 3 and 4 enantiomer, 1 and 3 or 1 and 4 diastereomer, 2 and 3 or 2 and 4 diastereomer. Clear? Now one more important thing here, I need to let you know the important part, that is the two terminologies which we use for such compounds. The two terminologies are trio erythrio pair. Okay, now what is this trio erythrio pair? See, when we have optically active compounds having two non-identical chiral carbon atoms, here we use the term trio and erythrio. What happens if the enantiomeric pair having two non-identical chiral carbons and enantiomeric pair, see, just like 1 and 2, enantiomeric pair having same atoms or groups on same side, it is called as erythrio. So 1 and 2, this enantiomeric pair, same groups on same side. Okay, so this will be your erythrio pair. And same groups, see hydrogen here and here, on opposite side, it will be called as trio pair. Clear to you all? So I can say 1 and 2 erythrio pair means same side I said, and 3 and 4 I said trio pair. Clear to you all? Understood this thing? See, this is also one more important terminology I have used here. Okay, so let me define these two terms. Erythrio. So erythrio pair, if I say erythrio pair, then I can say enantiomeric pair having two non-identical chiral carbon having enantiomeric pair having two same atoms or groups on same side. Okay, enantiomeric pair having two same atoms or groups on same side, then it will be called as erythrio pair. Clear to you all? For an example, I can say you can check here, it is having 1, 2, 3, 4, two chiral carbons, both are same groups on same side, it is actually erythrio. Okay, clear to you all? Note it down. So if I convert this, see, I am just changing here only. If I say erythrio, if I replace it by trio, then what I will say, enantiomeric pair having two non-identical chiral carbon having two same, no, it will not be same, it will be same atoms or groups on, yes, here you have to change, different side. Okay, so this is your erythrio. Now when I say pair, I means the mirror image of this as well. Okay, that will be erythrio pair. Now when I say the same compound having Br this side, hydrogen this side, so this will be your trio. Okay, and its mirror image and this one trio pair. So for trio pair, what I will say, enantiomeric pair having two same atoms or group on different sides. Clear to you all? Let me tell you one very important shortcut for this. You can use this shortcut TOES. Okay, just like SARC, if you remember, TOES, trio opposite side, erythrio same side. Okay, trio for opposite side, right? As you can see, trio opposite side, erythrio for same side. I think clear to you all. Okay, so dear students, I have explained you about trio erythrio pair as well. So we have completed this session. I think all must have noted this everything. If you have not noted, just watch the video once again, note it down. These all topics are very, very important for competitive examinations. Okay, see you all on our next video lecture.